Hello students, last time we were discussing about the blood cell formation. As we know, blood cells are of different types like RBCs, WBCs, lymphocytes and monocytes. So we will start from the process of formation of RBCs. As we all know that the process of formation of RBCs is known as hemopiosis or hematopoiesis, and we all know that stem cells are the producers of these blood cells. That is, all formed elements are derived from single population. There are many precursors of these blood cells like proerythroblast, myeloblast, lymphoblast, monoblast, and megakaryoblast. As we know, proerythroblast they develop into RBCs, myeloblast they develop into basophils, neutrophils, and eosinophils. Lymphoblast they develop into lymphocytes. Monoblast they develop into monocytes, and in the last megakaryoblast, which develop into platelets. Now we study about the blood cell formation. As we already know, hematopoiesis it is a process by which new blood cells are formed from their precursor stem cells. Each day, about 100 million new blood cells are formed from these precursor cells, and this process of formation of blood cells takes place in red bone marrow of humerus, femur, sternum, ribs, vertebra, and pelvic region. Red marrow actually they arise from new blood cells, that is hematopoietic stem cells. These red bone marrow contain immature erythrocytes. They remain in epiphysis, girdles, and axial skeleton, while yellow marrow or yellow bone marrow they are dormant type, that is they contain many fat cells. They store fat tissue and they are located in the long bones of adults. This there is a tissue framework for red bone marrow, and these are the reticulative connective tissue. Now we discuss about the cell lines, that is genesis of erythrocytes. As we know in diagram, you can see in diagram, in this there are different types of erythrocytes. So first of all there are proerythroblast, which are committed cells. They remain in reticulate site stage for 1 to 10 days, 2 days in circulation. They make up about 1 to 2% of all erythrocytes. If we discuss about formation of leukocytes, the granulocytes they arise from myeloblast, and monoblast they enlarge and form monocytes. If we discuss about platelets, the platelet forming cells from megakaryoblast they break apart into platelets. So here is the different type of stages or different types of red blood cells. These are red blood cells, and this is the formation of leukocytes, and these are the formation of platelets. So now the some giving some important information. First, blood cells that develop with the earliest blood vessels. Mesenchymal cells they cluster into blood islands, and late in the second month, liver and spleen take over the formation of blood cells. First of all, develop from the earliest blood vessels, and then from the liver and spleen. Bone marrow. Then bone marrow becomes major hematopoietic tissue at the organ at month 7. So there is a chart of formation of RBCs from bone marrow, which clearly demonstrate the production of RBCs. So here in the chapter of circulation complete, in which we discuss about the conductive system of heart, in which we discuss about SA node, AV node, bundle of His and Purkinje fibers. And then secondly we discussed about cardiac cycle and cardiac output, regulation of cardiac output. Then we studied about blood pressure, ECG, and factors affecting blood pressure. And in the last we discussed about blood components of blood and formation of blood components. So here we completed the chapter circulation. Thank you students.